Apoptotic signaling is a crucial biological process that dictates cell death in multicellular organisms. This carefully regulated mechanism is essential for maintaining homeostasis, shaping development, and eliminating damaged or potentially harmful cells. Apoptosis, often referred to as programmed cell death, is distinct from necrosis, which can result from injury or disease and typically leads to inflammation and damage to surrounding tissues. Understanding apoptotic signaling is key to advancements in medical research, particularly in areas like cancer therapy and neurodegenerative diseases. The apoptotic signaling pathway can be broadly divided into two main routes: the intrinsic and extrinsic pathways. The intrinsic pathway is activated by internal cellular stress signals, such as DNA damage, oxidative stress, or other forms of cellular injury. In this context, mitochondria play a pivotal role by releasing pro-apoptotic factors like cytochrome C into the cytosol. This event triggers a cascade of reactions that ultimately leads to the activation of caspases, a family of cysteine proteases that carry out the demolition of the cell. On the other hand, the extrinsic pathway relies on external signals, specifically, death ligands that bind to death receptors on the cell's surface, activating similar caspase cascades that prompt apoptosis. Caspases, the executors of apoptosis, are critical components within both pathways. They exist as inactive precursors and require cleavage to become active. Once activated, they target various cellular components, including cytoskeletal proteins and enzymes, leading to characteristic morphological changes, such as cell shrinkage, chromatin condensation, and ultimately cell fragmentation into apoptotic bodies. These apoptotic bodies are then efficiently cleared away by phagocytic cells, preventing inflammation and preserving tissue integrity. The regulation of apoptotic signaling is complex and involves various pro-apoptotic and anti-apoptotic factors. Proteins like BAX and BAD promote apoptosis, while others such as BCL2 and BCLXL serve to inhibit it. This balance between pro- and anti-apoptotic signals is essential for ensuring that cells respond properly to stress conditions and prevents undesirable cell death. Disruption in this balance can lead to pathological conditions. For instance, excessive apoptosis can contribute to neurodegeneration, while insufficient apoptosis can lead to cancer progression. In conclusion, apoptotic signaling is a fundamental aspect of cellular biology that plays a vital role in various physiological processes. Whether through the intrinsic or extrinsic pathways, understanding how apoptosis is regulated gives insight into the intricate balance of life and death at the cellular level. By unraveling these mechanisms, scientists can develop targeted therapies to manipulate apoptosis in various diseases, paving the way for innovative treatments in cancer, neurodegenerative diseases, and beyond.